All right, well, hello, college biology. So today we're going to be finishing chapter 5 notes, and I think we left off with how do organisms maintain osmotic balance. We talked about passive transport, how there's 3 types. Diffusion, simple diffusion, movement of high concentration to low concentration of any molecules. We talked about facilitated diffusion, which means that we move from high to low, but we are using protein channels or protein channels and carriers to do that. And then the third way is osmosis, where it's just the movement of water from high to low. So we have 2 sections left, and they're kind of short. And we're going to be looking at active transport. So what's the difference between active transport and diffusion? I'm going to talk about one type of active transport, the sodium-potassium pump, and then explain the energy behind all of that. So active transport is where we're going to move energy from, or use energy to move substances from low concentration to high. So you're climbing back up that hill, so you have to use energy. And we took notes on this, but there are 3 types of protein carriers. Uniporters, which means a single molecule moves through. Symporters, 2 molecules move through the same direction. And then an antiport, 1 molecule moves in, the other molecule moves out. So in opposite directions. Why is this not working? Okay, there we go. So active transport is really important because sometimes we have to move molecules against that concentration gradient, and this provides us a way to do that. We use energy in a direct or indirect fashion. So with the direct, here's an example of the sodium-potassium pump. So we have sodium-potassium pumps pretty much everywhere. So we have a low concentration of sodium inside, high concentration of potassium inside as well. So low sodium, high potassium inside the cell. And this concentration is maintained by pumping sodium out and potassium in. And it uses the energy molecule ATP. When ATP latches onto this carrier protein, the protein literally changes shape to help move the ions and prep for the next step. So here's the step-by-step of this. So step number 1, here's my cell membrane. We are inside the cell. This is outside the cell. So we want to have low sodium here and high potassium. So if we want low sodium, we got to move sodium out. So here's ATP. It latches on, and it undergoes an energy conformation after 3 sodium ions latch on. So you can see it's like this, right? And then energy conformation changes the shape of it. So now it's like this. And then the sodium ions are going to get released. When the sodium ions are released, these potassium ions bind to specific sites, and then it undergoes another shape conformation change here. Like that. And so literally the sodium-potassium pump just does this. Now, this pump can go pretty fast. It actually can do it 300 times per second. So it's like something like that, way faster, obviously. So yeah, sodium-potassium pumps. All right, here's a way where we use energy indirectly. So we're still using energy, but not directly for that movement, because ATP binds to that in the sodium-potassium pump. Well, here these are examples where energy is being used somewhere else, and then as a byproduct of it, we can move from low to high. So it's usually called coupled transport. Energy is released as one molecule moves down the concentration gradient, it's captured, and then we move a different molecule against its concentration gradient. So here I have a proton pump, which means it moves hydrogen ions. We're going to use energy here. We're moving from low to high. And now we have set up this proton gradient.
Hey,
You're fine.
You're not here.
Okay.
No. I'm just getting my stuff ready.
Okay. I'm going to be done in like 5 minutes.
Well, it takes me a while.
Okay. So
Let me tell you.
we've built up this high proton gradient.
What?
I'm talking to myself.
Oh. Okay.
So now we can move from high to low and not use any energy, but as a byproduct, we can move sucrose into the cell, and we need sucrose to make ATP. So here we use energy, and then we've captured that energy, and we can use it indirectly to move another molecule inside the cell. So, review question: which of the following membrane proteins does not rely on diffusion to function? Does not rely on diffusion. Your answer is D, the sodium-potassium pump. That's active transport. How are active transport and coupled transport related? And your answer is I want to say it's C. I need to check my answer. Yep, it's C. So it uses a concentration gradient established by active transport. All right, final section. Endocytosis versus exocytosis and how endocytosis can be specific. So bulk transport by endo and exocytosis. Oh, geez, Louise, that's a typo. It should be exo and endocytosis. So sometimes we have to use or move very, very large molecules, like large polar molecules across our phospholipid bilayer. And so when we want to bring substances inside the cell, we call that endocytosis. So material will enter through the use of vesicles. There are 3 ways endocytosis can occur, depending on the material that gets brought in. If the material is solid, then we call it phagocytosis, but we're still bringing things inside the cell. If the material is liquid, okay, then we call it penocytosis. So it's still endocytosis, but if it's all liquid, then it's penocytosis. And then sometimes for those specific molecules, we call them receptor mediated, where the molecule actually binds to specific receptors on that plasma membrane. So here's phagocytosis, where it literally just engulfs, surrounds it, traps it into a vesicle, and brings it in. Here's penocytosis, where we have some dissolved ions, and we're going to bring in some fluids inside the cell. So again, it just surrounds it or pinches in and brings it in. Over here we have receptor-mediated endocytosis. So these little green hooky things represent receptors that latch onto specific molecules. And when they latch onto enough, then a spring trap is formed, traps it, brings it in like a vesicle. So this is a diagram showing that receptor-mediated process. We'll start with 1. The ligand binds to the membrane receptor. 2. It migrates to an area called the clathrin-coated pit. So this is going to set up the spring trap. Okay, the spring trap is set, and it goes off and traps the ligand with the receptors. And then the cool thing is that these clathrin are released, and they get recycled, and they go back to the plasma membrane. And then depending on what these ligands are for, they will go to the lysosome, or they'll go to the Golgi apparatus for processing. But then eventually that vesicle returns back to the plasma membrane. So here's an example of receptor-mediated endocytosis, low-density lipoproteins. So LDLs, maybe you've heard of this in health, bring cholesterol into the cell where it is packed into the membranes. And there's this disease out there called hypocholesterolemia, where the LDL receptors lack tails, and so they never can fasten to the clathrin and spring the trap. So as a result, you end up with cholesterol, high cholesterol in your bloodstream, forming plaques that could lead on to other diseases like heart disease or a clogged artery or a heart attack or stroke or whatever. So that's a great example of a disease where your receptors never fasten to that spring trap, and now you have cholesterol floating around your bloodstream. So then people take drugs to help these receptors fasten to the clathrin to capture and bring in that cholesterol so your body can get rid of it out of your bloodstream. Now, the final way to move bulk materials is through exocytosis, and we're going to leave the cell. So these materials are going to exit the cell. And this is really important when cells build their cell wall. They actually form little vesicles that leave the cell and then become part of the cell wall. And then it's very important in animals to secrete hormones for signaling and growth and development, neurotransmitters, again, for chemical messaging to help you coordinate and move, digestive enzymes to help you break down your food, as well as a bunch of various other processes. So here's exocytosis, where our Golgi apparatus is packaging and modifying these proteins and slapping that address label on, and then it's just going to float to where it needs to go. And usually in the process of, like I said, in exocytosis, it's going to leave the cell. So you can see that the vesicle morphs with the plasma membrane and secretes the substances. All right, review question. Which of the following is not a mechanism for bringing material into a cell? That should be A, exocytosis, because endocytosis means bring it in, and peno means to drink it in, phago means to eat it in. But that is the rest of chapter 5 in a nutshell.